Hello, this is my presentation on CAR T-cell therapy for the first and prestigious cell and gene therapy class offered here at Mass Bay. So what is CAR T-cell therapy? CAR stands for chimeric antigen receptor, and this kind of therapy is a type of immunotherapy which uses immune T cells that are altered in the lab that are enabled to locate and destroy cancer cells effectively. Currently, CAR T-cell therapy is FDA approved to treat aggressive B-cell lymphomas in adults and B-cell leukemia in children and young adults. T cells are a type of white blood cell, also known as T lymphocyte or thymocyte. The role of T cells in our immune system is to help protect the body from infections from foreign substances, and they are also responsible for cell-mediated immunity. T cells are genetically engineered in a laboratory, so they have a CAR protein coming off of its surface, so they have a new receptor that they can bind to target cancer cells and induce cell death. The process first starts off with the collecting of the T cells from the patient, known as leukapheresis, where blood is drawn from the patient's vein into an apheresis machine, where the T cells are filtered. Next is the genetic modification of the T cells, where they are taken to the lab and modified with a retroviral or lentiviral vector encoding the CAR protein into the T cells to express the CAR on its surface. And once the lab has grown enough T cells, they are then injected back into the patient. The engineering of the CAR protein attached to the T cell includes a viral vector, in this case a lentivirus, that is inserted into the patient's T cell with receptors drawn to the CD19 cancer cell antigen. This then instructs the T cell to produce CAR proteins on its surface. The CAR protein consists of an extracellular domain, a hinge region, a cell membrane, a co-stimulation domain, and an intracellular signaling domain. One of the diseases that CAR T-cell is FDA approved to treat is acute lymphoblastic leukemia, also known as ALL. It is one of the most common types of cancer in children of the blood and bone marrow, where it affects the white blood cells, causing them to grow into lymphoblasts, which are leukemic white blood cells that cannot fight infections. This kind of disease is caused by changes in the DNA of immature lymphocytes, symptoms including enlarged lymph nodes, high fevers, easy bruising, bone pain, and bleeding from the gums. Some of the effects ALL has on an individual would be suppression of the immune system, where the lymphoblast could suppress the production of normal white blood cells along with the blockage of normal blood cell production, in which the overproduction of lymphoblasts in the bone marrow can lead to other bleeding disorders such as anemia from the decrease in normal red blood cells, white blood cells, and platelets. Other effects would spread to the central nervous system, causing headaches, seizures, and neurological symptoms, along with impaired clotting due to the decrease in platelets and increased risk of bleeding. This picture shows how the CAR protein attaches to the cancer cell and induces cell death. Once the binding domain from the CAR protein, also known as the scFv anti-CD19, recognizes and attaches to the CD19 antigen on the cancer cell, the T cells then cluster, sending signals to the signaling domains, which consist of the co-stimulation domain and activating domain. The CAR protein then activates the T cells to release cytokines, which are toxic chemicals to the cancer cell, inducing cell death. Shown is tisagenlecleucel, an FDA-approved drug under the name Kymriah, made by Novartis to treat relapsed advanced B-cell lymphoblastic leukemia for children and young adults to 25 years. There are certain side effects to this therapy, including cytokine release syndrome, B-cell aplasia, and tumor lysis syndrome. This is one of the clinical trials to Kymriah, showing that 8 out of 10 patients successfully achieved an MRD negative remission within 3 months of this treatment, meaning that no disease was detected in the patients treated with tisagenlecleucel. These are my references. I want to give thanks to the biotechnology program and my professor, Dr. Singh. I'm really excited to gain more hands-on experience in the research-based lab we have here at Mass Bay. Thank you for listening.